Hello, champs! Welcome back to our channel, your go to place to make biology super easy and fun. Today's concept is one of the most magical process which is happening inside your body, that is the translation. So, we are going to cover the translation in just 10 minutes. So, let's go ahead. What is translation? Translation is a process in which we use the information on the mRNA to build a protein. Okay, so how we are going to use the mRNA to build a protein, we are going to see in our today's video. Talking about the site of translation, the translation happens inside the cytoplasm and mainly, or we can say majorly, on the ribosome, the machine where your translation is going to happen. Now just meet our main components, or we can say the key players of our today's translation. We have mRNA, that is the messenger RNA, which is going to carry the codons. It is going to carry the codons. Codons are majorly of 3 letters, which are the codes and which are present on the mRNA. Next is your tRNA. This tRNA is acting like a delivery guy who is having the anticodon for the codes of the mRNA. Okay, the next one is your ribosome. This ribosome is the one who is going to read the mRNA. He is going to read the mRNA and he is going to attach the amino acids. Okay, we are going to understand what are these. And amino acids are the building blocks for the protein. They are going to act like a building blocks for your proteins. Okay, so these are your codon. If we have AUG codon, we have anticodon for this codon. For A we have U, for U we have A, and for G we have C. So such kinds of anticodons are present. So hope so you have understood the main components of the translation: the mRNA, the amino acids, the codon, the tRNA, the ribosome, and the anticodon. So mainly our translation is divided into 3 main stages. We have initiation, we have elongation, and we have termination. Talking about initiation, the ribosome is going to assemble at the start codon, means it is going to mainly come and going to attach at your start codon, or we can say the start region of your mRNA. In the case of the elongation, the amino acid side chain is going to grow. How it is going to grow, we are going to see in the further slide with beautiful diagrams. And in the termination, the complete protein is going to get released that has been formed in this translation process. Talking about the ribosome, in the ribosome we mainly have 2 subunits. This one is a large subunit, and this one is known as small subunit. Okay, so this ribosome mainly has 2 sites, we can say. One is the mRNA binding site, where your mRNA is going to get binded, and the other one is the site for tRNA binding. Okay, but for tRNA we are having 3 different sites. We are having 3 different sites. First one is your A site, second one is your P site, third one is your E site. Okay, this A site is the site where new tRNA is going to enter, where new tRNA is going to enter. Okay, this P site is the site which is going to help, or which is going to hold the growing protein chain. It is mainly going to hold the growing protein chain. Okay, also this E site is the site which is going to say tata bye bye, that is, it is a tRNA exit site. tRNA exit site, where your tRNA is going to exit. Here If I tell you more about this, here your A stands for amino acyl site, amino acyl site. Your P stands for peptidal site, peptidal site, and your E stands for exit site. Okay, to tell you more, one more important information, that is only your methionine, only your methionine, methionine, that is your start codon, is going to enter from the P site. Rest all are going to enter from A site. Rest are going to enter from A site. Okay, this might be clear. Now we are going to understand the mechanism. Now, talking about the initiation, in the initiation, what happens? Your small subunit of the ribosome gets attached. The region it is getting attached, it is your start region, or we can say here are your start codons. Okay, so here will be your A, U, G, that is your start codon. Also, then your large subunit is going to come. Okay, what will happen now? This AUG, this AUG is coding for methionine, right? It is coding for methionine. This is where your protein begins, means every protein is going to start from the methionine. A special tRNA which is having the methionine is going to come. See, for example, this is your tRNA. This tRNA is having a methionine amino acid and also the anticodons. For AUG, we have anticodon U, A, and C. These anticodons are going to come and going to bind with this codons. Okay, so now I will show you the entire process in the further slide. See, in the initiation process, what is happening? If we see this slide carefully, we are having AUG. Okay, in this AUG, just a second, let me change the color of the pen. We have AUG. These are your start codon. This tRNA has just come, or we can say arrived. It is having this amino acid, which is this amino acid. This is methionine. Okay, and this were your anticodons for this codons. Okay, see, you can see how this mRNA is coming. So the site where your start codon has come, or your methionine amino acid has come, this site was your P site. See, this is the initiation stage. You can see very well. This is your mRNA, this is your large subunit, and this is your small subunit. How this is happening? See, this is how the tRNA, the which has formed the chain. This is your next tRNA which is coming with the anticodons. Okay, so this is your initiation. Now let's go to the elongation. Talking about the elongation, what will happen? The ribosome, this large subunit of the ribosome is moving on this mRNA. This is moving on the mRNA. What is this ribosome doing by moving on the mRNA? It is reading codons. It is this ribosome is reading the codons. By reading the codon, what is this ribosome doing? By reading the codon, the tRNA is bringing the right amino acids. The right amino acids. Now, the ribosome will connect your amino acid with the peptide bonds. You can see this is the amino acid and this one was the previous amino acid. They are getting binded with the help of which bond? Peptide bond. Okay. Imagine I have made this arrow. This is your peptide bond. The chain is growing. You can see this chain, the growing chain. It is growing longer and longer and it is adding more amino acid. Okay. And as this amino acid has been delivered by this tRNA, it is going to exit. Okay. Then there will be next tRNA coming into picture. This is how your protein chain is been formed. You can see over here this blue. Then let me erase it. This blue one, we just saw this blue one over here. It has been delivered by this tRNA. Now this tRNA will get released. Okay, now we are very close to our last step, that is our termination. So this is your termination. Actually, what happens when this ribosome reaches the stop codon? Stop codon. Which is your stop codon? The UAA, UAG, and UG. The stop codons are also known as nonsense codon. As this ribosome will reach the stop codon, for example, this is your stop codon. As it has reached this codon, this stop codon don't have any codes. Okay, they don't have any codes, and therefore it is a signal that now the translation must be stopped. Translation must be stopped, and the protein chain has been released. You can see this is the protein chain which has been released. This is how your protein chain is also released, and your tRNA is also going away. Your ribosome is also going away. Both the subunits are going away. See, this small subunit, large subunit, your tRNA, your mRNA, all are getting separate out. Okay, when this ribosome gets detached, this is a sign that translation has been completed. Translation has been completed. Okay, so this is your last step, that is the termination. Now let me show you the lastly all the three pictures. This was your initiation. In initiation, how this tRNA are coming. This was your start codon, right? See, this is how your another tRNA is entering. In elongation, we saw they are forming the peptide bond, and then the tRNA is been released. How it is delivering its amino acid. Then we have shown that our peptide chain is formed, and lastly, this peptide chain gets away. Your tRNA gets away, and the ribosome, when detaches, this is showing that your termination stage, or we can say your translation has been ended. Translation has been ended. Okay, so this is your diagram. See, outgoing empty tRNA, this has just released its amino acid. This is your growing peptide chain. This both are bonded by the peptide bond. This is your mRNA, and this is your subunits of the ribosome. This is the tRNA which is coming. This is a very good diagram where you can understand all the steps. So to tell sequentially, see, I'll just tell you sequentially what is happening. First, your ribosome is getting binded. Ribosome is getting binded. After this, your methionine is getting binded. Okay, the tRNA with methionine amino acid is getting binded. Then other tRNAs are coming and they are forming the peptide bond, and then they are getting out by delivering the amino acid. Okay, and as this tRNA is going to UAA or UGA, that is your stop codon, it is going to stop the translation. So this was for today, and that's how our translation is going to end right here. So isn't it cool how your genes are turning into protein? So this was our video for translation in just 10 minutes. If you found the video beneficial and useful for you, please like, share and subscribe my channel. Give a big thumbs up, share it with your friends and hit the subscribe button. Thank you so much.